So I think we already were looking at some of the markers, and here we have a talk which essentially deals with the next generation genomic technologies for enhancing genetic gains in chickpea for ensuring nutritional and food security. The talk will be given by my colleague, Rajeev Varshney, who is the Research Program Director for Genetic Gains Program at ICRISAT. He has been involved with many projects, especially on sequencing. He has been involved with the genome sequencing of pigeonpea, chickpea, peanut, pearl millet, sesame, mungbean, adzuki bean, and I don't know, many more to come. He's a member of several academies and very well awarded. So we look forward to hearing more from Rajeev, and I'm sure you'll enjoy the talk. Thank you.
Okay, very good afternoon, and thanks a lot for the introduction. I would like to thank Maurice Moloney and organizers for giving us this opportunity, and thanks to all of you that you are still here in the last session to the last talk. So thanks a lot for that. So in this talk, I will be talking about chickpea, but before that, just to mention that I am coming from ICRISAT, and ICRISAT is one of the CGIAR centers. And our philosophy is that we believe all people have a right to nutritious food and better livelihood. So our institute's activities, they are perfectly aligned in the mandate of this institute, Global Institute of Food Security, as well as for the conference. And our mission is to overcome poverty, overcoming hunger, reducing malnutrition, preventing environmental degradation. So anyway, so like ICRISAT and many other international organizations, they are working for the international agriculture. And when you talk about international agriculture, majority of time this is done by smallholder farmers. So they have always this, they always remain under the poverty trap. So they don't have enough input, so they are unable to provide inputs to the farming. As a result, they don't have enough produce, so they don't have the surplus income to again to provide the inputs in the farming. So basically, they are always in the poverty trap. So it's already difficult to come out from this trap. And then when you talk about this international agriculture, then we know that, well, for feeding the 7 billion people right now, that agriculture is important, and this is another major challenge. And we have been listening this thing for last 2 days that, well, how we are going to feed the 9 billion people. This becomes much more difficult in front of the international agricultural research community when we know that, well, half of the world's population could face water scarcity by 2030. And majority of time when we talk about international agriculture in developing countries, then this is dependent on basically on the rainfall or like that. Otherwise, in general, their agriculture is in the rain-fed conditions. We were also listening for in many presentations about this global hunger index, and then things are that maybe around 800 million people, or 1 in 9 of the world's population, go to bed hungry each night. So this is another issue that, and there are many other issues, I'm not going to touch those issues, but in summary, if you talk about this food security, nutritional security, then we are having 3 important dimensions of hunger. One is calorie deprivation, protein deficiency, and micronutrient deficiency. For calories, nowadays, means in the whole world, we don't have the major issue of calories, for instance, because we are having good production of wheat, rice, maize, etc. When you talk about protein deficiency, and we have listened again from many presentations that if you talk about the developing countries, then the proteins is coming from the plants in majority of the countries. So proteins play a very important role, and because of those reasons, that pulses crop, they provide an important component of proteins to the developing countries. Because of the importance of those pulses, 2016 has also been declared as the International Year of Pulses. So this underlines the importance of pulses. Why pulses are very important, and we have listed some of these things here that good for you, and Word has presented very nice overview about lentils. So these crops, they are the critical for food basket, important source of plant protein, and they help address obesity and diabetes. Also good for planet because you can grow them in very drought prone areas, and also help improving soil fertility by fixing soil nitrogen. And very, very important for smallholder farmers because these are the crops which can withstand climate change and then can reduce risk for smallholder farmers. And also very important in terms of reducing the carbon footprint. For instance, for producing 1 kilo beef, you need 15,000 liters of water. On the other hand, you just need 4,055 liters of water to produce 1 kilogram of pulses. So there are many important things, and in terms of this carbon footprints also, that to produce 1 kilogram pulses, you just add 0.5 kg. On the other hand, 1 kg beef, you need to add 9.5 kg. So there are a lot of important things with the pulses. There are different kind of pulses, and Bert has presented about lentil. In this presentation, I'm going to talk about chickpea. So chickpea is important legume crop, and this is grown about 14 million hectare around 59 countries with about 13 million tons of total production. In the world map, you can see that India is one of the largest, or not one, India is the largest producer of chickpea, largest consumer of chickpea, and largest importer of chickpea as well. Countries like Canada and Saskatchewan play a very important role here, that Canada, Australia, they also grow chickpea, but this chickpea is exported back to India. And nowadays many other countries, as Ruth mentioned on the other day, in Africa also, like Tanzania, Kenya, Ethiopia, this chickpea production is increasing, and this is going back to India. So for India, this is important for the food security, and for many other countries, this is an important industry crop and also provide that revenue. So chickpea plays important role. But again, if you see the developing countries like Kenya, Ethiopia, Myanmar, India, so majority of time the crop is grown by smallholder farmers, and they are grown in marginal environments, so crop is exposed to a range of the biotic and abiotic stresses. If you see the production constraint, then drought alone is very important production constraint or yield reducer for chickpeas. So in general, the crop productivity is about 800 or 900 kilograms per hectare, so always less than 1 ton per hectare. On the other hand, yield potential is quite high. This can go up to 4 tons per hectare. And of course, there are different biotic stresses, but in this presentation, I will be talking more about this drought or so. Now we know that, well, a lot of genome sequencing is happening. Several crops, food crops have been sequenced, and I have just put few where ICRISAT played important role. So I've just mentioned that, well, several crops are being sequenced, including several of these pulses crop. But the thing is that how this is going to help to the breeding program or help especially smallholder farmers. In our opinion, genomics research needs to be applied in the breeding program, and for doing this thing, you need to link this genomics with the genetics to understand the basis of the trait or trait mechanism, and also with the different logy disciplines like pathology, physiology, entomology or so. And you need to bring breeding in the center. That's what we are doing at ICRISAT. So when you combine this genomics approaches together with different disciplines and translate the genome information in breeding, we call this approach as a translational genomics in agriculture. And we are doing work in this direction for last several years. But moving towards this aspect, you need to have some basic infrastructure. So for instance, you need to have the genomic resources, you need to have precise phenotyping platform, you need to have the markers associated with the trait. But until few years back, in the crop like chickpea, we did not have those resources. In terms of the markers also, we had only limited number of markers, not very good genetic maps. So what we have been doing that during last few years together with the partners around the world, and including from University of Saskatchewan, and in this particular project, Saskatchewan Pulse Grower also contributed. So what we did that we sequenced the chickpea genome, because earlier we had issue of the marker resources. We also have the issue of that genetic variation and in that elite gene pool, so we wanted to have the good resources. So anyway, in summary, we sequenced this genome and we generated large number of SSR markers. We also did a lot of resequencing, and we found several million SNPs. And this project, not only that University of Saskatchewan, but Agri-Food Canada and Andrew Sharp is sitting here. So they were also playing very important role. So there are many partners. But anyway, so in summary, we started to develop the resources. We had the genome sequence. We developed high density genetic maps, physical map, and combined the genome sequence with physical map and genetic map, so that in the case if you would like to move from genetic map, you can land on the genes. Or if you are having the information on the genes, you can go back to the genetic map. So we got these integrated resources. And right now situation is very good, because we got a range of the marker genotyping platform, because one size does not fit all. So for different applications, sometimes you need large number of markers, sometimes you need a small number of markers, and then depending on the cost benefit ratio, you can use any kind of marker. So good news is that we got the suite of the markers now in chickpea. We got SSR markers, and also Golden Gate SH for 1536 SNPs, Casper SH, and now we also have the genotyping by sequencing approach, and recently we have developed the CGIAR axiom arrays with Metrics company, and we have about 58,000 SNPs. So we got good marker system in this crop. When you talk about drought is very complex trait, but at ICRISAT, our crop physiologists and breeders based on the work for last 10, 15 years, they come up with the plan that while roots play important role for conferring drought tolerance, but root phenotyping is very complex. So one of our physiologists, Junichi Kashiwagi, established this system where you have these PVC slender, you can grow your chickpea plant, impose the drought stress, and after that you can take out these plants and then measure those roots, and by using these scanners, so you can measure different traits. And they also established the good correlation between the field conditions and here in this PVC slender. But this is a bit tricky situation, but then we kept on also evolving in the phenotyping, and now this is the system called Leachy skin system established by Vincent Wadage at ICRISAT, and here you can also move these cameras from these different pots, and they can measure the different kind of traits. So we also got this good phenotyping platform. In addition to that, we also have several good phenotyping platform for measuring the different stresses, Biotech stresses, etc. So in terms of those phenotyping, I think that we are also good. And good thing with these platform is that in a single go, you can phenotype large number of accessions or segregating progenies. So I'm just giving the summary. The good news is that earlier we did not have many markers associated with the trait, but now based on the work of several colleagues in collaboration with several partners, we believe that we have at least 50 different traits mapped in chickpea where we got the markers associated with the trait. In several cases, those markers have been used in marker-assisted backcrossing. In some cases, we got the diagnostic markers. So we are having the different scenario for the different traits. But in summary, that for breeding applications, we got the good resources, and you are interested in any trait, we can have the markers. We can go in that basically closely linked markers. Sometimes that can be issue of that phenotypic variation. So we have, but anyway, in summary, you are having the good genetic maps, QTLs. In some cases, QTLs have been validated. In some cases, need to be validated. So we did a lot of work in this direction. But in this presentation, instead of going each and everything, I will like to present a story about this QTL-hotspot and molecular breeding for drought tolerance, that how the genomic resources are being translated to develop the varieties with the higher yield. So in this context, what we did that few years back, and with the colleague, we have chickpea breeder, Dr. Puran Gaur, so he had these two different segregating populations, recombinant inbred lines, and here you can see on that on the left-hand side that these populations, they segregate very nicely for the root-related traits. But at the same time, not only for roots, and when we did the phenotyping of these populations, they segregate for many other traits, and which are drought tolerance related trait. So this was one of the set. The second is that we used the germplasm also. At ICRISAT, we have more than 20,000 accessions in our gene bank. But from this set, our gene bank specialist like Dr. Upadhyay, he has developed the different sets. And one of the set in the center, we call it core reference set. And this is having 300 lines. How we defined? So from these 20,000 accessions, we defined a smaller set of 3,000 accessions. We call it composite collection. Then we genotype these composite collection with 50 SSR markers. So based on the genetic diversity and their passport information, we came up with a smaller set of 300 lines. And we believe that these 300 lines, they represent the chickpea germplasm diversities. That's the reason we call it reference set. So we started to use those segregating population and the reference set, and we wanted to do the phenotyping, which is very complex and very tedious job, but nevertheless, we phenotype those collections, so 300 lines of reference set, and also about more than 250 lines from each population, so total 500 lines for the different root-related traits, which, and many of these also, these traits they are listed here, includes root length density, root dry weight, etc. And we have done this thing phenotyping in 3 different seasons. But then breeders are always interested in their traits, because they say, well, so what that you are targeting the roots, we are more interested in yield and related traits. So what we did, that together, not only at ICRISAT, but together with several national partners in India and Africa, where in Kenya and Ethiopia, we have phenotyped those segregating population for several of those yield and yield related traits. And you can see that we are having the data for maybe 2 to 7 different seasons. And and then in many cases, especially for yield-related traits, we did this thing, phenotyping at large scale. So when we had this phenotyping data and the first generation genetic map we had in the chickpea, we identified a genomic region in one of the population ICC 4958 and 182. And in this genomic region, we had the QTLs for more than 13 out of 20 drought tolerance related traits, which explain up to 58.2% phenotypic variation. So we don't call this region as a QTL, we call it QTL-hotspot because you got the QTLs for more than 13 traits. The interesting thing is that this QTL-hotspot was not present only in just one population, but was also found in the other population in the ICC 283 and ICC 8261 on the linkage group 4. So we were very excited about that one. And then when we did this consensus map, we found this region at the linkage group 4, and because we had the genome sequences, we wanted to move further that, well, what kind of genes are present or whether we can narrow down that region, because initially based on the SSR map, the first generation map, the genomic region was quite big, and this was about 30 centimorgans, but nevertheless, we had several SSR markers. But now as technologies are changing, and this is good that the conference name is Emerging Technologies, and genomics area is one of the important research area where technology are evolving very fast, and together with partners, we have been using these technology very quickly. So adoption of those technology in our genomics research is also happening. So after that, when we had this genome sequence available and we wanted to move down further that, well, what kind of genes we have, there is a new technology called genotyping-by-sequencing. Here you sequence the entire populations instead of doing the SSR genotyping, etc. And here this population was sequenced and we generated about 700 million reads, and we got about 828 SNPs. So you get this data and then you align with the genome sequence. Otherwise, earlier maps, they were taking several months to years to develop, but now by using this technology, we were able to integrate about more than 828 SNPs very quickly in a few months' time. And then if you go back to this QTL-hotspot, you can see that we were successfully integrated 49 SNPs in that region. Earlier, we had only few SSR markers. And then what we did that we used that genotyping-by-sequencing approach where you use some restriction enzyme, but then together with Dave Edwards from University of Western Australia, what we did that we sequenced the entire population at 1x coverage. So instead of genotyping-by-sequencing where you cut that DNA with the restriction enzyme, and when you use this approach, we got about 53,000 SNPs. And when you want to develop the genetic map based on these number of SNPs, it's really difficult. So what we did, we started to construct the bins. So we divided the chickpea genome in different bins, and we got about 1610 bins. And then this is the approach. And by using this bin mapping, we have developed the recombination breakpoint based genetic map. And here you can see the linkage group from 1 to 8, and there are two colors. One color represents one genotype, parental genotype, the other color represents the other genotype. So this is quite high density genetic map. And we had the phenotyping data on this population. So what we did that we did that QTL analysis once again on this high density genetic map. And here you can see on the CALG404, so linkage group 4, the QTL, which was very broad at that time point, because of the high density genetic map, this peak became very narrow, and also splitted this peak or the big QTL regions in two major peaks. And then and in addition to that, not only on the linkage group 4, we also found the QTLs for several other chromosomes, like for instance on the LG8, and this is for days to flowering, days to maturity, etc. So the beauty of these kind of approaches is that earlier you were taking a lot of time, but now you can go very quickly in the genomic region. And because sometimes this is also asked many times, some breeders they ask how genome sequence is going to be helpful for breeding. This is one of the approach that you can reach to the genes very quickly. And then so now based on these detailed information, I'm not going to present, but what we did that this QTL-hotspot has been splitted now in two parts, QTL-hotspot A and QTL-hotspot B. And we know that, well, there are at least 26 genes present in there. Then we wanted to associate with the phenotype. So what we did that in addition, we also developed a high density population or high resolution mapping population where we generated about 12,000 F2s from that nil, where we have this segment, drought tolerant segment, versus with where we don't have anything. So by doing these things, we have these 12,000 F2s, they were shown in the fields, and we screened about 6,000, and this is PhD student Deepa who screened this 6,000 lines with those markers which are present in those QTL hotspot, and based on these things, you can identify the recombinants. And if you see on the left hand side that, and then on the right hand side, this is the phenotyping data especially for the yield. So what you can see that wherever you have this red color segment on the first part, 113 kb, in those regions you are having the higher yield, and if this region become the blue color basically coming from the other parental lines, then you don't have that much yield. So we are believing that maybe some of the genes they are present from that region. So we are doing this kind of work where we would like to clone, identify the genes. This was from the segregating population, but now the other approach what we did, that we have this reference set of 300 lines as I mentioned, and they are coming from 33 different countries, and we sequence these 300 lines at 10x coverage. And after that, we have generated or aligned the sequence data with the reference genome, and we found about 5 million SNPs, 596,000 indels, 512,000 copy number variation, and we are doing a lot of analysis. I'm not going to discuss these things in details, but another thing is that sometimes we wonder that why we are having this narrow genetic diversity in chickpea gene pool, and from this analysis we can identify that there have been selection sweep which have been responsible for reduction of the diversity, and we can do this thing analysis based on the wild species sequence data versus with the cultivated, as well as from the land races versus the breeding line. So we know that genomic regions which might have undergone the selection pressure, so by using the new approaches like pre-breeding, you can bring this diversity back in the elite gene pool. Anyway, we also have the phenotyping data on these 300 lines for several seasons, as I mentioned earlier, and when we did the genome-wide association analysis, we found about 250 marker trait association for drought tolerance, and these all the association or the significant, I have included the significant association, and some of them where breeders are interested, like 100 seed weight, we found about 166 marker trait association. Out of them, 98 were unique, 30 were present in more than one season, probably they are more interesting. And if you go in details, then we found that at least 43 of those SNP, they are going on the linkage group 4, where we already had the QTLs and where we did this analysis. So this provide much more or well credibility or maybe validated further the value of that QTL hotspot. For yield related trait, we had the 22 marker trait association under drought conditions, 16 for that for heat, and then we also found that several SNPs they are associated with the function. So now based on these things, so this was based on 300 lines. Now we have started another project where we are sequencing 3,000 chickpea genomes, and in this thing, so we are collaborating with large number of partners, including with Bunyamin Taran from University of Saskatchewan. And then what we are doing, we are sequencing these 3,000 lines. In addition to that, we are also phenotyping all these 300,000 lines at 6 different locations for the breeding related traits. So we believe that that kind of sequence data will be providing highly or high resolution association. So that was about the genomics genetics. Now if we ask the question that how we are using this thing in the breeding program, then I just want to remind you about this QTL hotspot. So what we did, that we started to introgress this thing in that breeding lines. In the cases of the molecular breeding for disease, it's very easy that you are having 1 or 2 gene deficit or you just convert or introgress the disease resistant genes in the susceptible varieties, you can convert those lines into disease resistant. Drought is very complex trait, and this is not so easy that you just transfer one gene and you can convert the line in drought tolerant. But when I was talking with the breeders that we need to transfer the QTL hotspot in your drought sensitive line, breeders are very smart. They say, no, if your QTL hotspot is having some value, transfer this QTL hotspot in our drought tolerant line, and we would like to see if this QTL hotspot enhances drought tolerance further. So this is a very pragmatic approach, but for many times for molecular biologists, this become a little bit scared that, well, how this is going to happen. But anyway, so what we did, that we targeted these 3 different varieties. JG11 is one of the most important and drought tolerant varieties in India. No other variety could beat the drought, this JG11 in terms of drought tolerance. We also targeted Shafe and Kaktu. These are the varieties from Kenya and Ethiopia. And on the right hand side, you can see that we did this marker assisted back crossing. So at each generation, we did this just screening of the progenies with the markers, and we kept on moving to the next generation. So by doing these 3 back crosses and 2 rounds of the selfing, And because in the case of chickpea, we can have 3 generations per year, so we did this finishing the work of 1.5 to 2 years, and we generated those introgression lines. We did the phenotyping of those introgression lines in the rainout shelter. On the left-hand side, you can see that different root length variations. So the first line is JJ11, which was that recurrent parent, but other lines, they are that introgression lines, and you can see that in each of these lines, we were successful to enhance the root length density. Breeders asked that, so what about the yield? So what we did, that we did the multi-location field trials at the 6 different locations in India, Kenya, and Ethiopia. And here on the right-hand side, you can see on the bottom that now we got several lines which provide 12 to 24% higher yield than the elite varieties, including JJ11, and JJ11 is most drought-tolerant variety. If you introgress this thing in many other varieties which are drought-sensitive, then yield advantage is going to be higher. And this is the result from Kenya, where 13 MABC lines, they showed 17 to 47% higher seed yield and about 12 to 40% higher biomass. In the case of Ethiopia, we found several lines where we are having even 40% yield above the standard check. So this is one of the successful example where you can transfer that, enhance the yield under the rain-fed conditions by using the molecular breeding. And now some of these lines, they are going under, in India, they have a system called ACRIP, All India Coordinated Research Project on chickpea, and then this will be released as a variety in 1 or 2 years. So this should be reaching to the farmers. Now you can do these things for 1 or 2 traits or like those things when we have the QTL. So what is the next for that genetic gain? And very quickly in 2 minutes, I would like to finish this thing. So here I have put this formula for that basically genetic gain formula, and which is that genetic gain is directly functional to the selection intensity, selection accuracy, and genetic variance, and inversely proportional to the years per cycle. If we would like to enhance the genetic gains in chickpea, which has been very low, then what we need to do, we need to play with these different component traits. And one of the important thing is that how we can play with this selection intensity, because if you are having the traditional breeding program, not only in chickpea, in many other crops, especially in developing countries, this is the classical breeding funnel where a breeding program makes maximum 20 to 50 crosses. From each cross, you are having about 100 F2s, and they keep on going to the different generation, and they keep on selecting the lines which are useful for them. But by using this approach, the selection intensity is very low, selection efficiency is low. If you are not using the marker information, then you are not having the genetic variance, so the genetic gain is very low. Now, to address especially the issue of the selection intensity, our proposal is, can we generate large F2 population? Can we start to generate large screening nurseries or make large number of the crosses? You can do it, but then how you will be able to handle these things in the field? So you can, if you generate 50,000 F2s, then you need to have the large scale field, etc. So to address this thing, now what we are doing, that we are moving towards the genotyping, where we can genotype these progenies in $2 per sample. $2 includes the DNA extraction itself. And what we are planning to have, that we would like to screen these progenies with the diagnostic markers for must-have traits. And right now we are working with several companies, and we have reached to the conclusion that, well, if we would like to screen 10 markers, maximum 10 markers, with a set of the 1536 samples, we can reach to this thing. And we are working with the different CGIAR centers, with the Gates Foundation as well. So CEMIT, IRRI, Gates Foundation, and many other centers, IIT and SIP or so. So our target is that we are planning to have these $2 per sample. So our plan is, if you see, we would like to say, instead of 20 to 50 crosses, in each year, if you can generate 100 F1 crosses, from each cross, you generate at least 500 F2s. In your first year on an F2s, you will be having 50,000 F2s. If you can screen these population with those must-have traits, then you can bring down this number from 50,000 to 5,000 or 2,000, I don't know what. Then you can continue from F3 to F7 by using the single plant selection method. And in this manner, you will be having the higher selection intensity, you will be having higher selection efficiency, because you are using this time markers associated with the trait. And then you do not need to go every year or like this thing in the field. You can do this thing quickly in that lab. So you will also be basically reducing the years per cycle. So we believe that by using this forward breeding or early generation screening, we can move towards new breeding funnel in that, not only in chickpea breeding, but in the breeding program in many other CGIAR centers. So in summary, chickpea has become the genomic resources rich crop. Earlier used to be called orphan crops. Now we have large number of traits mapped using the different approaches. I did not present all of these approaches, and molecular breeding is becoming the routine now. I just provided the example of drought tolerance, but we are doing similar kind of thing for the diseases. Forward breeding is important now, as I mentioned earlier, but then one of the important thing is that in this era when we can generate large scale sequencing data, genotyping data, we need to use the analytical and decision support tools. And especially when you are talking about these developing countries, it's very important that we need to have good support from the national government, international government. In this work, we have presented the work and the contribution from several colleagues from ICRISAT and many organizations from all over the world, and then we had that financial support from several organizations, Gates Foundation, USAID, and including SPG, Illumina, and etc. In the end, I would like to announce this or advertise this conference. We are Intra-Drought 5th, which we will be organizing in Hyderabad from 21st to 25th February, and drought is very important for ensuring the food security. So if you are interested, please, we would like to invite you to participate in this conference. We'll be happy to provide any other information, and we'll be happy to answer any question. Thank you very much.
Very good talk. It looks like you're using all the molecular tools that are out there. The only one I didn't see that's more generally used at the companies is gene shipping, or taking a ship of a seed and then only planting the seeds that have the right QTL backgrounds, so you increase your throughput even further. Are there plans to do that, or thinking about it?
Thank you very much, and this is very important point. So basically, when I was talking about this early generation screening, according to our plan, still we have to grow the plants, and then you need to take the leaf, and then you go for that screening. And what the question is, whether we can do the seed chipping, because many companies, when you have the seed, instead of growing the plant, you can chip the seed, and then you can isolate, or you can do the marker analysis, then the remaining seed can be gone for the sowing. So what is happening that in our case, so in some species at ICRISAT we work on the 5 different crops, but in the seed size is very small, at least in the 3 different crops. So it's really difficult to move in this direction. And second is that we were working with some other, like even we talked about about this thing with Pioneer. So in some cases like sorghum, we can do it, but we have not reached to that stage, but this will be really important. We need to include this thing in our toolbox. Thank you very much. Okay. So I think that everyone is interested to close the session now. Avais is having question?
I also work on drought in potatoes and sweet potatoes. So you simplify drought. I'm interested to see, because there's so many scenarios of drought. You have intensity, you have frequency, you have timing, and the physiology of plant responds differently. So your hotspot QTL for drought, will it be true in all scenarios of drought?
So very good question. The point is that you are right, drought is very complex, and I did not simplify, or I did not say that this is simplified what we say that well. So roots are one of the important traits related to drought tolerance, and initially when we targeted the roots, so when we were doing this phenotyping, we did the phenotyping, but at the same time we also did the phenotyping for yield and yield related traits under the rain-fed conditions. So the good thing is that when we did the QTL analysis, we found the QTLs also for those traits in the same genomic region where we had this roots related stuff. Our physiologist, now Dr. Vincent Wadage, earlier we did this lot of work with the Dr. Jonuchi Kashiwagi. He also comes up with the same plan that well, and interestingly that physiologists, they also keep on changing those traits every after a year or 2 years that well, now this is the trait, this is the trait. So he says that now BPD, so basically vapor pressure deficit or like that one or some other, the water dynamics is important than the roots. This is great. So what we did that we also did, he did the phenotyping of this population for his traits. And interestingly, this is by chance that when we did this QTL analysis, those QTLs also went in the same region. So our philosophy is we don't claim that these genes, they are the drought tolerant genes. And we need to see that one, but definitely these genes or this QTL-hotspot is heavily associated to enhance the yield. And when you say that, well, what has been enhanced? Recent example have shown that, well, majority of time this is enhancing the seed size as well. So basically when you enhance the seed size, yield is automatically increasing. So maybe there are genes related to the growth, related to the vigor, and which are responsible for providing the yield. We need to see this thing. Yeah, thank you.
Thanking all the 3 speakers for their excellent presentation. And I think we covered quite a bit of ground on nutrition, starting from Eric, and then what lentils are capable of doing and what this technology can do. So let's hope this QTL hotspot becomes more hotter. And then, so thank you very much, and look forward to seeing you at the pre-dinner reception in about 45 minutes. Thank you very much.